This will be the second part of S1P2. This will be the organic or biomolecule section of it. The preceding one dealt primarily with bonds and functional group. So organic molecules are any molecule containing carbon and hydrogen. So anything with a carbon, for the most part, is organic. There are some exceptions like carbon monoxide and carbon dioxide and things like that, but for the most part, all of these molecules will have carbon and hydrogen in them. There are four what we call biomolecules. These are the large molecules necessary for life. Because they are organic, they're going to contain carbon and oxygen and hydrogen, and additionally some might have other things like nitrogen or phosphate along with the carbon, hydrogen, oxygen. So here's kind of a summary of the four large biomolecules. Biomolecules that we talk about are macromolecules, so they tend to be these larger structures. We can divide them into carbohydrates, lipids, proteins, and nucleic acids. Those would be the large ones. Functions of carbohydrates, primarily dietary energy, a little bit of energy storage, and also in plants, some structural support. There's a few structural things in humans like glycolipids, glycoproteins that are there for signaling and communication, but for most part it's energy. The monomer form of a carbohydrate is a monosaccharide. Mono means one, saccharide is sugar unit, right? Don't worry about this drawing here. We'll talk a little more in detail about those. The monosaccharides examples would be glucose and fructose. Disaccharides would be like sucrose. And then with polysaccharides like glycogen or starch. For lipids are a little different. To kind of review from last time, lipids are going to be consisting of a bunch of methyl groups. Lipids are great for long-term energy storage and hormones, especially the steroid hormones, for signaling and communication. Obviously, there are important components of lipids in membranes and things like that. There's a number of different types of lipids. The triglyceride is one type. There are other types as well. So we'll talk a little about triglycerides, and we see those, examples of lipids are fats and oils and steroids. Steroids we can use to make steroid hormones. Another macromolecule is proteins. Proteins are very diverse. They make up enzymes and structural storage and communication and important for contraction and transport and all sorts of things. Protein's basic unit is an amino acid. Okay, we talked about that last time. It's got a carboxyl group, an amino group, and then some sort of side group. Right? There's a bunch of proteins, right? Lactase is an enzyme. Hemoglobin is a protein that carries oxygen in red blood cells. There's a whole bunch of proteins, right? Your muscle's made out of protein and all sorts of things. So protein's very, very diverse in terms of their structure and function. The last macromolecule is nucleic acids. They're primarily for information and energy storage. All nucleic acids have the basic unit of a nucleotide. It always has a sugar, a phosphate, and some sort of base. And great examples of nucleotides would be DNA and RNA. However, that's a nucleic acid, right? The nucleotides would be like ATP or, thymine or something like that. So we'll look at those, briefly. So we'll go through these in a little more detail, all of them. This is just another way to organize them, things like that. When you make a larger molecule, we've been calling them macromolecules. Macro refers to big, and molecule, obviously, structure. We can take a short polymer, poly means many, so a couple units, right? And add a monomer, a single unit to it. And you do it with what's called a dehydration reaction. And off the one end of one molecule, you take a hydroxyl group, and the other one you take off a hydrogen. That comes off as water, and now we've added 1 unit to our polymer. All right. Sometimes the dehydration reactions can be called synthesis reactions or condensation water coming off, right? And synthesis is to make larger molecules, right? So we can make bigger and bigger molecules by just putting more and more links onto the chain, basically. Now, going the other way, we can break up a larger molecule into smaller ones through what's called a hydrolysis reaction. Hydrolysis means we have to put water in to break it apart, and those water end up back at the ends that we had before. Okay, so we can break a polymer chain up with hydrolysis. Okay. We'll come across this word lysis a bunch of times this semester, so learn it now if you don't know already. Lyse means to break apart. Right? So break apart. So hydrolysis is a reaction that breaks things apart. So little carbohydrates. Carbohydrates are known as sugars. Right? Monomers are single units. Those are again called monosaccharides. Mono means one sugar unit. All right. And we can take 2 monomers, 2 monosaccharides, glucose and another glucose, remove the water through that condensation reaction, right, and make a disaccharide. It's got 2 units now, called maltose. Okay, so maltose, that's a sugar. Now, you can do the same thing if you want to make sucrose. Sucrose is table sugar. You just take a glucose, do that condensation reaction with fructose, right? So remove a water comes off, and we end up with sucrose, which is your table sugar. And there's a bunch of other ones, and I'm not going to worry too much about, what 2 sugars we put together to make a disaccharide, and, what are disaccharides and monosaccharides. What's probably most important for us are understanding monosaccharides. And if you're going to understand monosaccharides, the one to understand is glucose, because we're going to come across that all semester. And then we can take monosaccharides and put them into 2 sugar units to become disaccharides. And we take disaccharides and increase those and keep on putting sugar on them until we get a polysaccharide. Now, there are a bunch of polysaccharides that we see. Okay, so here's examples of a couple. One is starch. Now, starch is an important food item for us. Okay, and all starch is just repeating units of glucose. So if you look at this, you see glucose, 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 all the way down. All right. Now, where do we find starch? We find it in plant products, right? So plants basically store their energy as starch. And so we can eat plants, get that starch, break off the individual glucose monomers, those monosaccharides, absorb them into the body and use them for energy or to build other things that we need or store them. Now, how do we store glucose? Well, let's say you're going to go for a marathon run tomorrow, right? So marathon's a little over 26 miles, right? You would probably want to prepare today by increasing the amount of carbohydrate energy storage you had. And one of the ways to do that would be to eat starch. And as we break up those sugar units and now we start to store them in our cells, we don't store those as starch. We store them as glycogen, okay? Glycogen is stored in our muscle, both skeletal and smooth muscle. It's also stored in the liver as well. Now, notice that glycogen is also repeating units of glucose. There are a few minor differences. This starch molecule up here, it's hard to follow, but it's one single long strand. The glycogen molecule, though, is highly branched. Okay? And so storing energy as glycogen has some big advantages. It's hard to explain right now because we're going to talk about it later towards the end of this section, but it has osmotic implications by storing one large glycogen molecule as opposed to hundreds of small glucose molecules. And so it has a few other storage implications as well that work out pretty nice. But glycogen is the way we store sugar in our muscles and liver. Now, take a look at cellulose. Cellulose, right, is really just repeating units of glucose. So in all three cases, we've got these repeating units of glucose, but cellulose is, very different. We get cellulose from the cell wall of plants, and the cellulose we call in our nutrition classes, right, fiber. And so one of the big things that I'm sure most of you know about fiber is it's not digestible. We can't break down fiber into energy, yet it's repeating units of glucose. So how can we eat starch, break it into glucose, right, store excess glucose as glycogen, and then go run a marathon and use that glycogen to help fuel the run? But cellulose, we can't do that with, even though it's repeating units of glucose. So how do we do that? Well, before we get into how we do that, let's talk a little bit about simple versus complex carbohydrates. Okay? So simple carbohydrates are primarily mono and disaccharides, and this has actually been a little bit controversial lately, where this is called the glycemic index, where eating simple carbohydrates, so mono and disaccharides, tend to raise your blood sugar quickly, and then it falls off very quickly as well. Where eating more complex carbohydrates, right? So I believe that's a bowl of brown rice, it looks like, right? Oatmeal, things like that would be, potatoes, sweet potatoes, all would be complex carbohydrates. And because your body has to break this down enzymatically, it takes a longer time to get that into the blood, so the blood sugar level does not rise as quickly, nor does it reach as high as peak, nor does it go down as fast, right? So foods with a high glycemic index tend to release a lot of sugar in the blood very quickly, while ones with a lower glycemic index take a while longer. Okay, so that's why we tend to prefer the complex carbohydrates over the simple ones from a dietary standpoint. Although the concept of glycemic index and its usefulness in nutrition has been questioned recently. So let's get back to the idea of cellulose. So, we can't break down cellulose, but, cows should be able to, right? They need energy, and pretty much what do they eat all day? Grass, and grass is pretty much made out of cellulose. So how do cows do it? Well, the answer is they cheat, okay? They can't break down cellulose themselves. However, they have microbes in their gut that allow them to break down cellulose. So they have these, basically, small microorganisms that break down the cellulose for them into sugar, and then they can regurgitate it and put it in a different stomach and, be able to use it to make energy. Also, before we leave, termites do the same thing. All right, so that's kind of the different ones. Oh, one last kind of idea before we go on to lipids. There is another polysaccharide called chitin. All right, so it's spelled Chitin. So it looks like it's chitin, but it's pronounced chitin, and that's what makes up shells of crabs, for instance. And what's interesting about chitin is it's just repeating as a glucose with a little bit of nitrogen. So by just adding a little nitrogen to that kind of cellulose type structure gives us a shell of a crab. So you can see, how, chemically similar all those molecules are, but how functionally different they are as well. One last thing I probably don't think I did because I don't have a slide of it is let's talk a little bit about carbohydrates before we go. Carbohydrates always have the general formula CH2O. So they're going to be made up always of that formula, CH2O. So as an example, if I said, well, glucose has 6 carbons, right? How much oxygen and hydrogen does it have? Well, it's always going to have that formula as a monosaccharide. So if it's got 6 carbons, right, it's got twice as many hydrogens, so you have 12, and it's got 6 oxygens, okay? So if I said, okay, ribose, which is the sugar in RNA, is a 5-carbon sugar. How many hydrogens does it have? Well, you know from the formula then that it's going to have twice as many, so it's going to have twice as many as 5, so 10, right? So it's C5H10, and the oxygen is going to equal the number of carbons, O5. So it's got that basic formula. Because it's got those hydroxyl groups on it, right, with the oxygens, it's going to have a charge distribution of the molecule. So it's held together by polar covalent bonds, so it's going to be a polar molecule. Glucose has an energy equivalent of 4 calories per gram, right? The fundamental unit is a monosaccharide, and our primary use of carbohydrates is an energy source, although there are some structural and communicative functions as well, but those are relatively minor. All right, so let's look at lipids. Now, lipids are structurally very diverse. Lipids also have the basic formula CHO, but notice the difference. When we talked about carbohydrates, we said that was always going to be CH2O in the case of monosaccharides. For lipids, they're going to have those methyl groups, right? Those non-polar carbon-hydrogen methyl groups. So they have, very few oxygens within a lipid, okay? Lipids tend to be, because of those methyl groups, we call non-polar, or sometimes they call them hydrophobic. Okay. Since we're kind of talking about structure, let's go through those parts that we just did for carbohydrates and compare them. Lipids, because of the methyl groups, are non-polar. I just said that. Lipids are calorically the most dense biomolecule we have. We get 9 calories per gram. Remember, it was only 4 calories per gram for carbohydrates. The fundamental unit is a fatty acid, which is sort of a lie because not every lipid is made up of fatty acids. But for our purposes in this class, the purposes of exams and checkups, things like that, the fundamental unit of a lipid is fatty acid. As an example, though, steroids are an example of lipids that are not made up of fatty acids, but actually derived from cholesterol. Right? So fatty acid is our fundamental unit, and fatty acids are part of what we call triglycerides, part of phospholipids, which help make up the lipid bilayer in our cell membrane, and again, do not make up steroids, but steroids and cholesterol and waxes even are also subclasses of lipids that we can see from a functional point of view. Lipids primarily function as energy storage. We can store a ton of lipids as energy. It's good for insulation, and it's important for communication, especially if you're a steroid, right? It makes up the cell membrane, so it's protection. So those are kind of the introduction to the lipids. Examples of lipids, beeswax is what you see there. butter, and then, Barry Bonds, because he was a famous baseball player who took steroids, which is, again, a lipid. So lipids can be divided up into kind of two groups: fatty acid-based lipids and non-fatty acid-based lipids. Fatty acids, right, have many methyl groups. So all these carbons here, C, 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 all the way up. Notice they're just a hydrogen. Here's the methyl group at the end as well. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 carbons long, okay? 15 of which are the methyl group. And then one carboxyl group. Remember, that was the acidic group when we talked about functional groups, right? So there's my carboxyl group at the end. So you say, okay, is this going to be polar or non-polar? And oftentimes in humans, we're not really good at estimating things sometimes. And so we sometimes look at what's different without realizing all the things that are similar, I guess is the best way to put it. So you look, oh look, it's got a carboxyl group. It's acidic. It's going to be polar, right? You say, yeah, that is an acidic group right there, and it is a polar group, and that's one of the 16 carbons. The other 15 though are non-polar, right? And you could probably guess, if you've got 1 part polar and 15 parts non-polar, that the overall molecule is going to be what? Non-polar. So fatty acids tend to be non-polar. Now, we can take a fatty acid and stick it onto a glycerol molecule. A glycerol molecule is a sugar alcohol, and it's going to be 3 carbons. So it's got 3 carbons, and it's got the hydroxyl groups, right? So that's a sugar alcohol, okay? And when we do that, we take this fatty acid, you don't have to know it's palmitic acid, okay, and stick it on to this oxygen, right? We form 1 part of a triglyceride, okay? So putting 3 fatty acids on a glycerol molecule, it's called a triglyceride. And triglycerides are important ways we store fat. So do you want a long-term energy storage molecule to be acidic? Would it be good? No, we don't want to be acidic, because remember we mentioned before a little bit about pH, and blood pH has to be normal, right? And then sort of answer the other question, number 2, how many fatty acids can be attached to one glycerol molecule, right? Well, we've got 3 carbons, so 3 can be that, and you form a triglyceride. So here's an example of what we call fat, triglyceride. So here's our glycerol head in green. Here's our fatty acid tail in yellow. Notice that the fatty acids are different lengths and also different shapes. Okay, so we'll talk about that. But generally speaking, most fatty acids in the human body are somewhere between 8 and 36 carbons long, and most are in the 16 to 24 carbon range. Right? We'll see when we talk about metabolism, we believe there's a good reason why most of your fatty acids in the human body are an even number of carbons. And when we use this for energy, what we do is we break off the carbons into 2 carbon chunks. And so if you had an odd number of carbons in the fatty acid, when you got to the end, we'd have this 1 odd carbon that you couldn't make into energy because you're supposed to have 2 carbon chunks. So it makes sense that most of our fatty acids are even numbers, right? Also, looking at the difference when they're bent, it's the difference in not in linear. It's the difference between saturated and unsaturated fatty acids, which we'll get into in a minute. So here's a triglyceride. It's a different way to represent it, right? A glycerol head and a 3 fatty acid tails. Okay, so that's where the tri comes from, the 3, and the glyceride refers to the glycerol molecule that we originally had. What's interesting about the triglyceride is this part in gray here, right? That's called the head. That's going to be polar because it's still got the oxygens, and the oxygens here are still going to be electronegative, and they're going to hold on to its more share of its electrons, so it's going to be a polar head. These tails, they're pretty much all methyl groups, and so this is going to be a non-polar molecule. When you have a molecule that has polar and non-polar parts, it's called amphipathic. So as an example, an animal that lives on both land and water, like a frog, you call an amphibian. So that amphi part, just put pathic on it. So an amphipathic molecule has both polar and non-polar ends. And the triglyceride is a great example of that. All right. Is a triglyceride acidic? All right. So not acidic anymore because we no longer have a hydrogen to donate. So it's not acidic. And what's the layman's term for triglyceride? Right? Well, fat, right? And, technically, fat tends to be a solid at room temperature and oil tends to be a liquid at room temperature, although through modern chemistry, we've changed that idea. But generally speaking, that. And when you look at adipose tissue in humans, right, that's how we store fat, is triglycerides. Now, fatty acids, as I mentioned before, can be saturated or unsaturated. If you have a saturated fatty acid, right, it doesn't have any double bonds, okay? And so it means it's saturated with hydrogen, and those tend to be solid at room temperature, like lard or butter. And saturated fats that are solid at room temperature tend to be generally not as good from a health standpoint for people, or at least some studies have shown, many studies have shown, that saturated fatty acids can lead to increased heart disease and things like that. Unsaturated fatty acids have double bonds, and all those double bonds are cis. We'll talk about what that means in a minute. And those tend to be liquid at room temperatures. Okay, and it's because the double bonds cause these bends, and then if they're bend, the fatty acids can't stack in together to become solid. Okay, so a saturated fat, right, is going to have all these linear, right, saturated fat methyl groups, and there's no double bonds here. And because they're all linear, you can stack them very tightly, and that becomes a solid. Okay, so you don't have to know this, but stearic acid is what they show there, and that's a saturated fatty acid. Now, if we look at something like olive oil, right, olive oil has these unsaturated fatty acids in them. And when you have these unsaturated fatty acids, it causes that bend. And because we have this bend, we can't cluster the triglycerides together as easy, so that's why it ends up in an oil. Okay. So we can kind of compare them. The saturated fats are linear, they have no double bonds, so they're straight, and you can compact them, and that's a fat at room temperature. And, the unsaturated fats have double bonds, which cause a bending of the molecule. They can't pack as tightly together, and those tend to be oils at room temperature. But years ago, scientists decided, well, wouldn't it be nice to have one behave like the other? And so what we've done is we've taken these natural cis isomers. Cis means that the X's are on the same side, to a trans isomer, which means the X's are on opposite sides. Okay? And so when we do this, we can take a oil, make it a trans isomer, and now we can straighten them out. And when we straighten them out, we can make them just like saturated fatty acids. And for a while, we thought that was a good thing. But then we decided that numerous studies showed that people that had a lot of trans fatty acids in their diet ended up with higher risk of cancer. So now we don't see trans fatty acids, especially in the U.S. Years ago, they outlawed them, but gave companies like, I don't know, it was like a 10-year period before they had to be out of our food chain. And most products ended up getting rid of them very quickly because the public responded by not buying trans fatty acid foods. And so they had to be, changed over to non-fatty acids foods. So they had to figure out other ways to make them taste good and give you the whole texture and everything that you wanted without changing them too much. Here are a couple examples of non-fatty acid-based lipids. You might recognize these words. Estradiol is a form of estrogen, and testosterone is the male sex hormone. Notice they're very similar in structure, right? One, testosterone has an extra CH3. Remember, that's a methyl group, right? And then over on the other side, instead of having a hydroxyl group, they just have that double bond O here. But, this basic structure background here, right? These four ring structure, that's indicative of it was originally a cholesterol molecule. So cholesterol is what we use to derive our steroids and steroid hormones from. And it's a non-fatty acid based lipid. Just, one more idea on looking at lipids. We said the average glucose, not the average, a glucose molecule by definition has the chemical formula C6H12O6. And just to kind of give you an idea of, it's always got that CH2O ratio. Well, fatty acids in humans can vary tremendously, but as an example, the average fatty acid in the human body has a chemical formula very similar to C55H1O4O6, right? So it's got very few oxygens relative to all the carbons and hydrogens, which makes it have many methyl groups, which makes it nonpolar. Okay, proteins. Before we go on, I didn't talk about this either, sorry. Let's go back to our fatty acids. So here's an example. Let's see. I like this one better. So here's an example of a fatty acid, right? We have actually 3 of them hooked up to a glycerol. So this is our triglyceride, right? Which we also call a fat molecule. Well, if we take one of these away and put a phosphate in its place, then we make what's called a phospholipid. What's interesting about this phosphate, it's going to pop over to this side and try to stay as far away from these nonpolar fatty acid tails, right? Because the phosphate, remember, it's got that big -2 charge. It's about as polar as you can get a -2 ionic charge. And so let's be a phospholipid. Where do we find phospholipids? Well, that makes up the phospholipid bilayer within the cell, right? So that's where those come from. So let's go back to talk about proteins. So proteins are functionally the most versatile of the biomolecules, right? We said fats, energy, a little bit of signaling, some insulation, a few other things. Glucose is energy, a little bit of signaling and some structural stuff in plants, not really so much in humans. Nucleic acids we haven't talked about yet, but that's for information and energy transfer and storage, right? And then everything else I haven't talked about for the most part comes under the purview of proteins, okay? So functionally, they're by far the most diverse molecule. Now, proteins, remember, are made up of amino acids, and we looked at the amino acids before, right? The amino group is the NH2. The acid is the carboxyl group. That's the C double bond OH. C double bond O, OH, right? It has a central carbon with a hydrogen, and then that side chain. It's the side chain that's variable. Okay, so proteins, because they're made up of amino acids, always have carbon, hydrogen, oxygen, and nitrogen in it. The side chain of the amino acid can be all sorts of things. Okay, we looked at the simplest amino acid. The R would just be a hydrogen. That would be glycine, right? And then you can see this nonpolar amino acid called leucine. Right? What makes leucine nonpolar? Well, its side chain is a methyl group, a methyl group, a methyl group, and a methyl group. So even though the amino acid part, right, that basic structure that's in every amino acid, right, are polar things, this tends to behave as a nonpolar amino acid because it's got all these methyl groups. Well, sometimes it can be polar and not ionized, where the oxygen and hydrogen, right, we're going to get this non-polar covalent bond here, right? And sometimes it's ionized, where we grab an extra hydrogen on this amino group at the end, right? And you've got all sorts of things in between, okay? There are 20 naturally occurring amino acids that we incorporate into proteins, right? And there's a whole bunch of other amino acids that occur in nature, but we don't put them into proteins. So as an example, some of you might have heard of creatine. Creatine is a supplement that people take that helps transport energy out of a mitochondria for use of ATP in the cell. She does a few other things too, potentially. But it's an amino acid, but we don't incorporate it into a protein, right? So not every amino acid in nature is incorporated into proteins, although there's a group of 20 that for every organism we've ever studied, it uses those same 20 amino acids. A couple other things about proteins before we go on. So the basic structure is the amino acid. It's always made up of carbon, hydrogen, oxygen, and nitrogen. Overall, they tend to be polar proteins, right? They tend to be polar. They have 4 calories per gram, so 4 calories per gram. The fundamental unit, we said, is amino acid, and they're the most varied in terms of the functional part of the biomolecules, right? And so remember, I mentioned earlier that, you know, it's the details that are important in terms of class. So if you looked at some of the videos before in the earlier portion of the class, and if you read the things, you know, on the introduction to the lecture material or the lab material, you know, I make a point about details as a point where, you know, students struggle sometimes. And not just knowing the details, but being able to demonstrate them and apply them to a novel situation. So, you know, here's where the details become important. If I asked you, what is the most structurally diverse type of biomolecule, right? You'd say, oh, there's a whole bunch of different ones in terms of lipids. Remember, I said lipids have the fatty acid based and the non-fatty acid based, and a wax is very different than a, you know, steroid, which is very different than a triglyceride, right? Well, if I asked about functionally, right? So the key word is functionally. What's the most functionally diverse amino acid? That would be, most functionally diverse biomolecule. That would be a protein. Okay, so proteins are very, very functionally diverse, and they do a whole bunch of different things. Examples of that, right? They could be structural. They could be enzymes, they could be storage molecules, they could be transport molecules, they can be communication molecules, they can be receptor molecules, they can be carrier molecules, they can be movement or contractile molecules, they can be defense molecules. And those are just, you know, a list, you know, pretty much off the top of my head. So, you know, they're very, very diverse in terms of how they function. All right. So proteins are formed by sticking amino acids together through that dehydration reaction we talked about before in general. And we just stick the carboxyl end of one amino acid to the amino end of another amino acid, remove the water, and now we get a growing structure. The bond that holds them together is called a peptide bond. And when you start to elongate this chain, it becomes what's called a polypeptide. Poly, many, peptide, referring to peptide bonds. So a polypeptide is made up of a structure that has many amino acids stuck together by peptide bonds. Okay? Depending on the function and the size and a few other things, and really who's arguing about it, amino acids linked together by peptide bonds eventually form proteins. There used to be a requirement that a protein had to have 50 amino acids to be officially a protein, but now we know that it doesn't necessarily have to be that way. So again, the amino acid structure, right? We have that amino group, that nitrogen, we have that carboxyl group, the carbon with a double bond oxygen and OH, and then that side chain. You don't have to memorize these. Here are the 20 amino acids kind of grouped into nonpolar, polar, and electrically charged groups. So we can see there's some of each. Right? We're going to use amino acids in chromatography and separate a couple of these out within a solution. So we'll kind of look at that at some point in time. Kind of see how that works and measure their RF because each one will behave potentially different because of their side groups. And again, if we link a carboxyl group with an amino group of 2 amino acids through that dehydration reaction, we get a peptide bond, and now we get a dipeptide, di meaning 2, and we can keep on going together. And once you get, it's not even 100, but once you get it long enough, you get a polypeptide. A protein is made up of 1 or more polypeptides. As an example, hemoglobin actually has 4 polypeptide units to make up one portion of the protein. Here's an example of all the different protein functions: enzymes, structure, storage, transport, hormones, receptors, contractile, defense, right? All examples of different proteins. So that's why it's functionally so diverse. When we have amino acids together into a protein, the order of amino acids, does it matter? Absolutely, right? So here's an example of hemoglobin. Normal hemoglobin has 146 amino acids stuck together, okay? But in someone with sickle cell anemia, they have a sickle cell hemoglobin molecule, and it's called a point mutation, okay? And what happens is in position number 6, okay, glutamine, right, is replaced by valine. Okay, so if we come over here, right, when we have these different amino acids, sorry, glutamic acid's one's replaced. So here's glutamic acid, right, and here's valine. So notice glutamic acid is an acidic, electrically charged molecule. Notice the side chain right here, right? And valine, right, is these methyl groups that we talked about before a little bit. A little different than leucine, but very similar. So coming back over here, if we look at number 6, it should be GLU, which is glutamic acid or glutamate, if you want to call it that. Remember the 8-inic acid, similar. And it's been replaced just in one of these 46 by valine, and that causes the sickle cell trait in terms of blood. So scientists have traced, at least the last time I read, 18 different independent genetic changes within hemoglobin in certain populations. Sickle cell trait, if you have a one copy from one parent, not both, gives you an advantage if you get malaria. And it has to do with the organism that makes you sick from malaria. Part of its life cycle is in a red blood cell. And so by the red blood cell sickling, it decreases that intensity of the disease, it's believed, and so you're more likely to survive. The problem is it's very painful if you have the sickle cell disease itself, and it does no good in the United States because we haven't had any cases of malaria in the U.S., for a long, long time. So, it's evolutionary-wise, though, we've developed that trait in populations where they are exposed to the mosquito that causes malaria. So the primary structure is very important, and it's sort of like the order of letters within a word, right? Spelling out the word God, G-O-D, right, is a very different meaning than if you reverse the letters and spelled out dog, right? Even though it's the same letters, the sequence of the letters matters. So that's called the primary structure, and that matters, okay? The secondary structure has to do with the way the protein folds. And there's two folds. We're not going to worry about it. One's called the alpha helix. The other one's called the pleated sheet, or sometimes known as the beta pleated sheet. So how these fold up on each other, right, is determined by the primary structure. And just as a review, if you look, you see an oxygen and a hydrogen there and a little dashed line, right? And so those are held together by hydrogen bonding. Remember, we said the three-dimensional structure is held together by hydrogen bonding. Then you go into a tertiary structure and what they call quaternary structure. So these are all degrees of three-dimensional shape. And we're not going to worry about this except to say primary structure is a sequence of amino acids, and it matters, generally speaking. Not all the time, to be honest, but it matters. And then the three-dimensional shapes are the secondary, tertiary, and quaternary structure. Okay. Last category, nucleic acids. We're not going to look a lot at nucleic acids in this class, but nucleic acids are always going to have carbon, hydrogen, oxygen, nitrogen, and phosphate. And the reason being is they have sugar, so that's the CHO, right? They have a phosphate, that's the P, and a base is always what they call a nitrogenous base. So it's got nitrogen in it, so that's got nitrogen. So that's why that's always have CHONP for nucleic acids. Because of this, especially because of the phosphate, see these two big negative charges here, they're going to be polar. We do not break down nucleic acids for energy. So it has 0 calories per gram, although we do break these down for raw materials in the food we eat. And anytime you eat anything natural food, it's going to have DNA in it. So we'll be able to break those down. And the fundamental unit is called a nucleotide. You can see it here, nucleotide. So just like we saw before, if we stick monosaccharides together, right, we get a polysaccharide. If we put fatty acids on a glycerol, we get a triglyceride. If we put amino acids together with peptide bonds, we get a protein. If we put nucleotides together, we get a nucleic acid. Okay. Now, the sugar in DNA is called deoxyribose. In fact, that's where DNA gets its first name from. D is deoxy. The sugar in RNA is called ribose. They're both 5-carbon sugars. And that's why RNA is called ribonucleic acid. Okay, so these are 5-carbon sugars, not 6. The nitrogenous bases are different for DNA and RNA. Right? Common between both DNA and RNA are the cytosine, thymine, right, and adenine and guanine, right, are in DNA, and then in RNA, the uracil replaces the thymine. So the cytosine, adenine, and guanine are in both, and thymine is in DNA, and uracil is in RNA. So those are the nitrogenous bases. Now, if we take that adenine, right? So if we take that adenine, oh, let's not go to the adenine yet. Let's see. Okay, I'm a step ahead of that. So if you look at the structure of the nitrogenous bases, we see what are called pyrimidines and purines. We're not going to worry about it, but they just have, similar structures. All the pyrimidines are just one ring, and all the purines are two rings. And when they have that structure, that allows us to form that double helix. Okay? So that allows us to form that double helix. Now, if we take that adenine, right? So this is just an adenine molecule, and put 3 phosphates on it, we end up with ATP, adenosine triphosphate. I think all of you know that ATP is the energy currency of the cell. It's what the cell uses to energetically cause cellular reactions, cellular things to happen, allows us to contract proteins, allows us to move molecules against gradients, things like that. So this ATP is a very important molecule, and it has to do with energy, right? So there's other forms of energy, like we're going to talk about NAD and FAD, and those are nucleotides that have to do with energy transfer. And again, RNA and DNA are really just information storage, right? So information storage and energy transfer are what important in terms of the nucleic acids, right? And here's an example of how ATP works. So if we take this adenosine molecule with its 3 phosphates, chop off that last phosphate. Now it's not attached to anything, so they call it an inorganic phosphate. So they give a little I here. And we're left with adenosine diphosphate. So triphosphate, 3 phosphates, take 1 away. We have 2. That's called adenosine diphosphate, ADP, and that gives us energy. What's interesting is this works in both directions. So if we take energy and phosphate, stick them back onto an ADP, we can make ATP again. And where do we get that energy from? Biologically, from the food we eat. All right. So, ATP is regenerated all the time. So chemical energy, right, from our food molecules makes ATP, and then the energy for cellular work goes back to ADP plus PI. So as long as we keep adding energy, right, we're going to be able to go through this ATP cycle. All right, here's a nice biomolecule summary. I like charts and tables because it gives you a lot of information. It shows you the biomolecule, the calories, the basic structure, whether it's polar or non-polar, what the functions are, and what the basic units are. So I'm not going to go over them because we did, but you can look at them on yourself, and that's certainly a good one to know. So that concludes the section 1, part 2 chemistry section. Don't forget that you're also responsible for section 1 PX1 and section 1 PX2. That's the basic chemistry and the basic cellular stuff that we're not going to have a lecture video for, as you're supposed to learn on your own, but it shouldn't be a learning on your own because it's supposed to be in the prerequisites that you took the class for. So don't forget those.